Hello everyone, I'm Angeline Kirbai, Assistant Professor, Department of Biotechnology, St.Josephs College of Arts and Science for Women, Hosur. Today we are going to see about microorganism and classification of bacteria. Microbes are minute unicellular organisms that are invisible to the naked eye. They are also known as microorganisms or microscopic organisms. The term microbes is used to describe several different life forms with different sizes and characteristics. A few of these microbes include bacteria, fungi, protista, viruses, and archaebacteria. Classification of bacteria. Bacteria can be classified into various categories based on their features and characteristics. The classification of bacteria is mainly based on the following: shape, staining, mode of respiration, temperature, and mode of nutrition. Classification based on shape. First one is coccus shape. These bacteria are spherical or oval shape. On the basis of arrangement, cocci is further classified into Diplococcus. That means coccus in pair. Example, Neisseria gonorrhoeae. Second type is Streptococcus, coccus in chain. Example, Streptococcus salivarius. The next, Staphylococcus, coccus in bunch. Example, Staphylococcus aureus. The next, Tetrad, coccus in group of 4. Example, Micrococcus species. The next shape is Bacilli. These are rod-shaped bacteria. On the basis of arrangement, Bacilli are also further classified as Coccobacilli. That means shape is a combination of cocci and rod. For this type, example, Brucella. Then Streptobacilli, chain of rod-shaped bacteria. Example, Bacillus subtilis. Then comma-shaped bacteria. Example is Vibrio cholerae. Then spirochetes. They are spiral-shaped bacteria. Example, Treponema pallidum. The next classification is bacteria on the basis of staining. First, gram-positive bacteria. Cell wall of this bacteria is composed of peptidoglycan layer only. Example, Staphylococcus and Streptococcus. Gram-negative bacteria. Cell wall of this bacteria is composed of peptidoglycan and outer membrane. Example, E. coli and Salmonella. And classification on the basis of mode of respiration. First one is aerobic respiration. The bacteria are which can respire only aerobically. That means it respires in the presence of oxygen. They are generally get killed under anaerobic condition. Example, Bacillus subtilis. The next respiration is anaerobic respiration. The bacteria that grow in the absence of oxygen are called anaerobic respiration. Anaerobic bacteria does not have the ability to detoxify oxygen. Example, bacteroides and E. coli. And facultative anaerobes. The bacteria which generally respire aerobically but switch over to anaerobic mode of respiration if oxygen becomes deficient. Example, Klebsiella. The next type is obligate anaerobes. The bacteria of this category respire only anaerobically. They generally get killed under aerobic condition. For this type, the example is Clostridium botulinum. The next classification is based on nutrient. First one is autotrophs. These bacteria can create their own food from inorganic compounds. Autotrophic bacteria are further divided into two groups. First one is photoautotrophs. Photoautotrophic bacteria uses carbon dioxide as their carbon source and convert it into carbohydrates by using sunlight. It contains bacteriochlorophyll and bacterioviridin pigments in their photosystems. Example, Helicobacterium. The next type is hemoautotroph. This type of bacteria uses inorganic sources to create organic compounds in the absence of light. These bacteria do not contain pigments and only carry out the dark phase of photosynthesis. For this type, examples Nitrosomonas, Nitrococcus and Heterogenomonas. The next type is heterotrophs. These organisms that obtain their nutrition from other organic sources which can be living or dead. Heterotrophs are further divided into two groups. First one is photoheterotrophs. Photoheterotrophic bacteria use light as their energy source but cannot use carbon dioxide as their carbon source. Instead, they obtain their nutrition from organic compounds in the environment, such as carbohydrates and fatty acids. Example, green sulfur bacteria and purple bacteria. The next type is hemoheterotrophs. Chemoheterotrophs obtain their energy and carbon source from organic compounds such as sulfur, carbohydrates, and lipids. Example of this type of bacteria is decomposing bacteria, fungi, and protozoa. The next classification is based on temperature. First one, mesophiles. In this type, the bacteria grows in 20 to 45 degrees Celsius. Example, Staphylococcus aureus and E. coli. The next, thermophiles. Optimal growth temperature is between 45 to 122 degrees Celsius. Example, Thermus aquaticus. Then psychrophiles. They are cold-loving microbes. The growth is minus 15 to 10 degrees Celsius or lower. Example, Arthrobacter species. The next type is hyperthermophiles. In this type, the bacteria grows above 80 degrees. Example, Pyrolobus fumeri. The next classification is based on the spore formation. Spore forming bacteria. Those bacteria that produce spore during unfavorable conditions. These are further divided into two groups. First one is endospore forming bacteria. Spore produced within the bacterial cell is called endospore forming bacteria. Example, Bacillus, Clostridium, Sporosarcina. The next type is exospore forming bacteria. In this type, spore produced outside the cell. Example, Methylocyanus. The non-spore forming bacteria. Those bacteria which do not produce spore is called as non-spore forming bacteria. Example, E. coli and Salmonella. That's all about classification. Thank you.